Hello, everybody. Welcome to today's webinar on closing the gap in reflective PPG SpO₂ testing. So my name is Kyle Griffin, and if for some reason you can't hear me or anything, maybe post the question in the Q&A to let us know. So today, we're going to just quickly go over what the current existing test methods are that we have encountered in the field with customers and the testing gaps that those create. And then I'll hand it over to Stefan, who is our FAE, and he is an application expert in PPG applications, and he is continuously working closely with our customers. He'll go over a quick PPG testing workflow and introduce some of the core principles and a workflow flowchart. And we will also do a quick demo of the solution. After that, we'll have a Q&A session, and you guys are welcome to stick around as long as you'd like and ask questions. So for the Q&A, feel free to ask any question throughout the webinar using the Q&A tab. At the end, we will, Stefan will try to answer as many questions as he can. And if we can't get to your question, we will still answer it. We'll just do a follow-up email to everybody to share that answer. Okay, so I'm going to briefly go over the current test methods that are typical for reflective PPG testing. So first is gray card testing. And what that is a gray card testing checks whether the PPG hardware can emit light and receive a reflected signal. So your LED transmit light off of a color card or a gray card, which then reflects that light, and you can test whether your PD can receive it. So it's very useful for basic optical path check, signal level, and SNR verification. And we often see it as a production line hardware test, because it is a cost-effective and quick test. And the limitations are, it does not simulate physiological PPG behavior. It cannot verify SpO₂ or HR algorithm performance. And it's a hardware level test only. It's not a full system validation. Next, we have tissue phantoms. So a tissue phantom is a physical fake tissue model designed to imitate how the light signal is absorbed, scattered, and reflected by skin and blood. So here you can see there is one second. So here, you have a mold of a finger, and then you embed a 3D printed U-shaped channel, and you build a fake finger with the mold, and then that creates a sort of a fake finger that you can then test your SpO₂ device with. So the strengths are that it mimics tissue-like optical absorption and scattering, and you can evaluate the interface between the LED and PD arrangement and the skin. So the main limitations is it's a very expensive and difficult to build consistently. It's hard to control and standardize across setups, and it is usually a more research-focused method. It's not currently a commercial purchase-ready approach. So that means you generally have to build this test setup yourself in-house. And just to show you how complicated it can be, here is a recent research paper in 2025 that goes over the current method. So you use a pressure pulse generator to send a pulse wave through a fluid reservoir, which then transmits that through a U-shaped channel that's embedded within the fake finger, and that simulates the movement or the pulse for your DUT to pick up. And you'd also have to, since this is a do-it-yourself sort of testing approach, you'd also have to develop the pulse wave as well. So for human testing, these are just quick volunteer tests using your prototype, and often you will pair it with a reference SpO₂ device. So this is where you ask your colleagues or even yourself, or maybe a subject group to put on your DUT. It's fast and easy. It uses real human signals, and it allows for a quick reference device comparison. And the weaknesses are it's not controlled or repeatable method. It's mostly normal or healthy condition people that you're testing on, unless your colleagues are all very sick. And the edge cases, so that makes edge cases hard to test repeatedly. And even if you use an FDA 510K cleared reference device, the accuracy is still limited because the FDA allows for plus or minus 3% accuracy. And what about clinical studies? Well, we work with our customers to make sure that the clinical study is truly for the final validation of regulatory claims, because that is not the ideal place to discover performance gaps, as that could mean, rework, narrowing of claims, and delays. So our solution, PPG Simulation, helps close the gap with controlled, repeatable system-level testing. So what it is PPG Simulation uses controlled optical signals to test how the wearable device responds across defined SpO₂, heart rate, PI, AC/DC, and R value conditions. So just to give you a simple diagram, on the top would be your DUT here. And on the bottom is our simulator. And there's a separator that separates the PD and your LED. And the LED will transmit a pulse pattern, a light pulse pattern to our PD, which will receive it. And then we will send a simulated reflection back to your PD with the biosignal for your PD to receive. Okay. So just kind of diving in a little bit more, down here is an example of your DUT, and it will be sending out a light pulse pattern. Here is our simulator's PD, and it will be receiving that pattern, and it will be in sync sending out a PPG simulated reflected waveform, right? So here is our simulated reflection from your the pulse pattern that you're sending out. So we are simulating that, we are sending that out, and then you will be receiving that with your PD. And this piece here, is an example of a segment of a PPG waveform. So what are the strengths of this approach? It's full system level testing. You're not just testing hardware. It enables repeatable edge case testing. It makes it easier to compare design and algorithm changes, so you can have before and after testing, and as well as good documented evidence of the changes that occur after you have a design or algorithm update. And it allows you to systematically test the full claimed performance range of your DUT. But like any test methods, there are limitations. It can't replace clinical trials. It doesn't do SNR testing. It doesn't reproduce human tissue optical behavior. And since you need to align the PD, your PD with our LED and our PD with your LED, sometimes we need a test fixture to align the light paths. All right, so next I'm going to hand it off to Stefan, who will dive into the principles. Just give us maybe 20 seconds to help do that transition and do the setup. So just hang in there real quick. Thanks.
All right, thanks, Kyle. And hello, everyone. I'm Stefan. So next we can have 25 minutes, and here is the map. The first 5 minutes is a zoom out, the 8-step industry validation journey. And then we will anchor the bench testing as what ISO say it is, and the 3 engineering metrics we're going to hit before clinical study, and then let's have a deep dive on SpO₂ bench test and a demo before we close with a takeaway and Q&A. So before we dive in, one question: how confident are we in SpO₂ testing before sitting a human subject down in clinical studies? Over 200 wearable companies we work with, they have done some grid card testing, maybe some early human wearing, or they think the algorithm is working, but they have not been able to severely verify repeatability and test what happened at low perfusion or confirm if the R curve is behaving the way they expected. And this is the gap Kyle mentioned earlier, and that's what today is about. Okay, let's zoom out. Every serious wearable team that's going to bring a wearable to the market go through some version of these 8 steps in roughly this order. Number 1, define a claim. Before we touch any hardware, we need to answer what vital to measure, what body location, what population, adults, pediatrics, starts into what conditions, rest, exercise, or sleeping. And this is definition will really constrain everything that comes after it. Get it wrong here, we will spend months of validation the wrong things. Yeah. Number 2 is the basis SNR. So it's like the loop test from the DUT LED through a reflection interfaces, maybe just a board or gray card, and reflect back to the PD. So it's a loop test the system against the internal noise, a noise flow, and it works at the basic level. Reasonable SNR or no gray optical failure, and that's what we can see from this test. And it's fast, it's simple. It should catch obvious hardware issue before we go further. But this is important. It tells you nothing about SpO₂. And then number 3 is where we use the SpO₂ signal simulator at bench testing. So we start verifying hardware response, SpO₂ behavior, repeatability under various conditions. You can control PI heart rate waveform morphology. You can build a real engineering data set. I'll go deep on this just a few minutes. And then we have a pilot study, not clinical accuracy, just because, yeah, it's a evolving human. Yeah, it's not. But for everything else, does the sensor actually stay on the wrist? Does motion artifact destroy the signal? Is the raw PPG quality good enough for SpO₂ calculation? That's where we get the real-world optical coupling data. Yeah, this is a step we really, get into the world from our lab. And number 5, we're coming back to the bench testing. Why? Because now the bench test becomes even more powerful. We take the real PPG waveform, collect in step 4, import it into the simulator, and replay them. Now we can, tune algorithm, run regression tests, stress each case, or with some real physiological signal. Yeah, and do it thousands of times without a single additional human subject sitting in. And then, good, we're gonna lock it and no more change. This is a version we will take into the formal clinical studies. And that's where we mostly, familiar with. Yeah, calibrate our device with human subjects or reference oximeter as secondary standard in ISO AS/ISO 1-2-61. And XDD, yeah, they allow 2-way. Invasive way and non-invasive way. And this study is, of course, expensive and effectively constrained and time limited. And this is not we want to discover the algorithm and I'm not working. So after authority approval, we are allowed to go big. We start scaling up production, bringing wearables to the end user. Then we can access thousands of real waveforms as devices are worn. From if there's a critical update in the future, we can feed those data back to the regression suite at step 5 over here, to irritate the verification and use them for next generation algorithm development. And they become a data set for your company for years to come. Actually, we have a case from the customer in Swiss. And the FDA advised them to add a minor feature when the signal is low in PI, when the algorithm is not so confident about the reading. So when they detect such a signal, weak signal, and they should just raise alarm. So to do that, they did two things. First, they find a weak signal from those previous clinical trial or the human pilot runs, and then also use simulator to provide the low perfusion, which is like low AC signal, and see the threshold of the algorithm and when it's going to raise the alarm. And that process happened like this, and going back to the FDA examination again. Right. So functional tester cannot replace clinical study, but accuracy is mathematically impossible because achieving repeatability and reproducibility first. Yeah, so those two is kind of like make the electronic stable and readable. So based on those, we can really go for accuracy. So now we can look at the step 3 and 5 a little bit more closely. The functional bench test actually happened twice. So once early in development, and again night before the design is freezed. And this is not a coincidence. The bench is the backbone, is what allows us to do a severe repeatable engineering work at a stage where the human study are not yet appropriate or not yet needed. If we skip step 3 and step 4, or start step 5, we can order pay very low attention on those two. We're going to the clinical study blind. And that's a very expensive place to discover any problem. So that's anchor bench test in the standard. So the Oximeter standard ISO 80601-2-61 and version 2017. And now we have 19, and now we will have 25. So this haven't been updated. It still stands that there's annex specific about the functional tester, FF.5. It defines the functional tester as a device that uses simulated optical signal to interact with duties LED and PD, providing a stable and repeatable reference signal. 3 things we can remember from the slide is number 1 is repeatable. The human vary, the bench doesn't. You can run the same signals that 1,000 times and get the same results. And it does not replace clinical study. So the ISO is pretty clear on this. The functional tester is to serve building confidence before clinical study. And third, it covers the full optical path. So you can verify the whole signal process chain. So from the signal that come from our simulator, you will get into a PD and then FE, and then become the raw data in ADC counts, and then DSP, and eventually algorithm. Yeah, so this is where we can verify the whole signal chain. And so if there, for example, the FE misbehave, we will see it there. So here's the practical picture. Things in orange is where duty is on its own in clinical study. So before the duty enter this part, you need to be well prepared. So for the reference, you can even use the invasive ABG test, blood gas test, or the non-invasive way using the 510K approved as a secondary standards. And this is the device to submit, and both will get signal from the human subjects. So the signal source is human subjects, and then the computer reading. If A close B, and that's great. That's the usual way. But even we can choose non-invasively. It sounds less expensive, but it still takes time and money. So before we get there in a button, this is where the annex FF.5 mentioned a functional tester. From here, we can build a confidence. Yeah, so before we go into either invasive or non-invasive, so you can inject a stable signal to both device, the device to submit, also the reference. So the both will read the same signal in turns, and if the A almost equal B, and that's engineering confidence, not clinical proof. Yeah, it's because it's outside the orange parts, but confidence matter to the engineers. And this is exactly why annex advice envision. We are not replacing clinical study. We're making sure when we're walking through the study, we already have strong reason to believe our device works. And in the origin context, it mentioned a test finger like this. But for the wearables, we also provide the reflected interface as here. Yeah. the illustration Kyle mentioned in the first few slides. So now we're going to zoom in. We're going to walk through the actual WhaleTeq SpO₂ bench test workflow. First step, this where we will see the ACG software. So step 0, just the basic understanding. Yeah, the most important thing is about the understanding the reference R curve, or you can call it bench R curve. And there's a common misconception that people think they should program their calibration curve into here, because see, we have some upside down button, we have column to enters, but please don't do that. The equation in the ACG software is not any duties internal calibration curve. Is that benchmark curve is mimic the shape of the duties algorithm for the purpose of the simulation. We will unveil the reason why it's different. But so let's focus on the AC100 about its red PI equation and IRPI equation. And you can see we have ACDC for red and ACDC for infrared, adjustable under your control. So here we have a quite useful function. It will get you started just in no time. And where this is SpO₂ table, so we can first enter ACDC, the parameter under your control, and let our duty to feel what the signal is about. Yeah, it's just the AC superimposed on the DC in the red channel, and the AC superimposed on the DC in the red channel. But we don't know the answer, so let the duty let us give us the answer. So this is repeatable testing. Send it this 1,000 times, you should get the same reading. That's the repeatabilities. So do it at a different level. So we're gonna tune the red AC component in a different row, but keep the rest of the same, and see. So we understand a little bit science that the red AC have a negative relation to the SpO₂. So when I increase it, I can see less SpO₂ present. And then we can plot the table to another coordination is SpO₂ versus R value. And what is R value? R value is the PI ratio of R and IR. And each PI is the red AC divided by BC, and IR AC divided by DC. So we're gonna plot these rows to the new coordination, like here. So this is each red dot is the test row, and we will also plot a regression curve to have a minimum distance to each one. So you can choose linear or parabolics. And now here, this is the reminder, and we're gonna unveil the differences from the internal calibration curve of your device. Is that the light path is different, because the calibration curve of the duties is between clinical trial, and the interaction happen between human skin and your sensors. But in the simulation on the bench testings, from our simulator sources through the lens and the fixture that will hold the tool, hold the instruments and the duty together, that's also the distance. And then eventually your sensors. So those two are whole different paths. That's why numerically different for those two equations. Right. So this is the end of the learning process. So what we do until now is to help ACG100 to know how to interact with this DUT by just trying an error and figure it out, kind of reverse engineering. And the end, now the learning completed, and we're going to start the actual testing now, because the game changed. We used to change the red AC. Hope we still remember, and to explore different SpO₂. Now, following the curve just created, so we can select it from the drop-down menu, and the corresponding red DC will be auto-config. So this is a number like 13.55. There's no normal people will think of this number, try to enter this kind of words, unless it's an auto-config from such a curve. And we can output the signal, and you will see 88 just on the DUT. So if you will run again and again, and a different the saturation level, and this is the repeatabilities. If we run the same signal over multiple DUT units, like in the in the QC pilot runs or even mass production, and that's the reproducibilities. And we also have the auto sequence functionality in the GUI over here, and this can sweep from the SPO to 100% down to 70 on your device, so you can immediately catch all of them and let your backend to jot down the reading at each measurement, and then we can compare against the expected curve and flag any deviations. So before I leave the bench test workflow, let me be explicit about the two practical use. So number one is the preclinical accuracy. You can say preclinical engineering confidences. So before that, we will mount your device and the 510K devices and on the simulator in turns. So for example, we create this table based on the secondary standards, and then I bring this here and put the duties on and let it get send signal. And does it shows just like here, just like the reading in this column? And if it's not, yeah, please note the tolerance and see in which region. For example, from 90 to 100, usually we try to maintain 1% accuracy. From 90 to 82%, down 70, they can be 3 to 6%. So here, for example, I can see the 2% is undesirable, and we can figure out, yeah, so this is where you can start vacation on that part. Right. So instead of these, if they track closely, we have strong engineering confidence going to the clinic. But is there any systematic bias at a particular saturation level? We just found a calibration problem before we are charged for a failure clinical study. And completion number 2 is the production quality control and for the reproducibilities. So once we have established a reference R curve, we use the golden units. Yeah. So now all the new units in batch to come is gonna follow the quality of this golden one. Yeah. So the data set we create for this golden, this is exactly the data set we're going to use on the production. But of course, consider the productivity is the priority number 1, and maybe let's get 2 or 3 out of those rows. And every unit that comes off the line gets tested against the 2 or 3 rows, and reading in the tolerance cross the batches, and it passes in the production. If any device that fall outside of tolerance, flag for offline investigation. And through the SDK, you can run this, automatically, after this integrated to your MES systems. Yeah, right now I'm showing a GUI, but this concept can be realized in, automation and in the integration of your EMS systems. And then we're going to have a small demo on each case. Yeah, and the duty we're going to use is the max inverse design 103 from the ADI. So this is the duty, and you're going to send pulse, at this, constant strength. Try to get a sample back from the patients. Go through the hall of the fixture. So the fixture is to interface both, and in the middle we have a wall. So between after the wall, there's another hole. So through the hole, we get into the PD window of the PPG modules. And when the PD is triggered, our LD is gonna turn on, shoot it back, but along the samples, yeah, we're gonna adjust our LD intensity in order to sketch a PPG curve on your PD, your AFE. So through another hole is coming back to your PD. So it's how it works. And if there's a more complex in your LED PD layouts, we can talk about it after after that or offline and reach your sales representative. We actually 2 years ago we have a webinar about how to accommodate the complex LED PD properly use spread like in the circle, spread like in a square, and we also have a solution just for that. So that before we start the demo, we will show the 2 basic robust tests for the wearable SpO₂ device. So this is a tool index. The first is the SpO₂ robustness against PR swift. Okay, so we're gonna keep the same SpO₂ level like 75. Hey, this is not usual. We don't usually get this from human being unless human is in critical condition or in clinical trial where we already spent money. So now we can easily create 75 rating on DUTs. And now we're going to only change the pulse rate to check whether DUT's SpO₂ reading remaining stable when the heart rate change. Because now the frequencies get faster. And let's see if the FE can handle that. So right now I'm open up the back end of the maximum, and now it's at 60. It's already we're going to show 75, right? But because if under the 80 is already have 3 to 6%. So now it's in. All right. You can say it's kind of stable. So let's speed up a bit to 180. We play. I see immediately the new signal caught, and then it become much faster, and see if it's still okay. Yeah, it's pretty okay. It start there, in 1%, 2%, but it coming back to 1%. And let's see what happens if we go the maximum 300. It's about like 5 hertz, and new signal cuts, and then he's gonna process this, and still in 1. Oh, now even like higher, but still in 1 or 2%. So it's pretty robust against the PR split. All right. So next we gotta look at the the SpO₂ robustness against the PI split. So we're gonna decrease the red and IRAC level. You can discover that. We're gonna keep it for doing this. We can keep the same SpO₂ level. Why? Because from the formula, if you can really go back, we can go back to here and see if the red AC and the IRAC decrease proportionally or increase proportionally, it doesn't affect R value. So when R value is not changed, SpO₂ is not changed. Yeah, so this is the mathematic trick I try to do using the PI swift. So right, so we're gonna start with 12, 10. Yeah, that's what we know. Like normally we do this, bigger number, normal numbers, and then we're gonna divide by 2, divide by 2, divide by 2, and keep the same constant DC level. And the PI is actually getting lower from the 0.15 and 0.125. This is pretty low for human being. We don't usually get this, signal before clinical trial. But right now it's pretty easy to realize on the bench test. So right now I'm gonna go, for example, in this table, I'll open it up. I found, all right, 95. So for 95, I should enter 12 and 10. All right, regardless the 400. So now I'm gonna program 12 and 10 and 1000 DC. All right, and let's see what we'll get. So usually we get last time we 75, right? And give it some time to stabilize it. And maybe we don't use the 300s. So right now it's reaching 95. So I mean, as already confirmed, the PR speed doesn't matter. Yeah, but for this, we can really use 60 like normal. And now I'm starting to decrease the PI by decreasing the red AC and our AC proportionally. Now this is a decreased, and let's see. So it's still within 1% from 95, and that's great. And then let's decrease 3 and 2.5. Right. Now it's become even smaller, the amplitudes you can see. And when its amplitude gets small, the noise level will noise flow start being dominated. So you can see some flaws in the waveform, but the backend can process that in the digital process. All right, so it's a little bit off it from the 95, and now we can even investigate more by tuning down Oh yeah, I'm sorry. This should be 1.5. Right. So now both red and infrared AC component gets pretty small. But we can still see the pulsatile signals. Yeah. So we can look at it, we have a zooming on the red channel and zooming on the infrared channel. We can see, yeah. But eventually so now the SpO₂, start performing unexpectedly. Yeah. So there's where we can discover if the SpO₂ reading is robust enough against the PI sweeps. And we can also define the range of the sweeps and see that, we can, characterize this index. Right. All right. Also, this wrap up of the demo. And then we can land the aircraft on the takeaways. So first, the backbone is mandatory. So the functional bench test at step 3 and 5 is a low-bearing spine of the verbal validation. And the step 3 and step 5 will satisfy the 3 functional tester definition and the standard. So our solution is standard-driven. And from the order of operation, we need to understand the repeatability and reproducibility is the foundation before we chase the clinical accuracy on a human being. So this can be realized, this can come true, and the capability for those two can be realized on a bench test. And the methodology in our proposal is not only just help you understand the optical path. We are doing the open loop testing instead of the closed loop testing in the gray card. The function can help you fill out the SpO table is in the exploration of SpO₂ reading versus AC changes. And then from the result, we can plot the table to the correlation, so we can access the parabolic R curve, the bench R curve. Yeah, the bench R curve, not the calibration curve in any DUT algorithm. Eventually, we can make the good use of the bench R curve to simulate different saturation level. From there, you can start to evaluate the performance of the DUT. Yeah, so eventually, our solution doesn't not only serve the R&D purpose, it also help with the quality control and productions. Yeah, so for the automation, yeah, or even the ability to integrate to your MES systems. Yeah, thank you. So this is pretty much the content for today, and we are ready to take questions from you guys.